DNA is great, but we don't actually directly use it for most of what we need to do. If you've got one beautiful set of blueprints, you don't want to hand it out to people to take to the field to get dirty and banged up. They can get damaged and lost. And plus, you might need it for someone else. So you need to make a copy. You need to make a photocopy of it. And this effectively is what RNA does. So let's scroll back up to RNA. RNA, if we look at its molecules, is very similar to DNA. You know, guanine, adenine, cytosine, they're the same. Only difference is it incorporates uracil instead of thymine. And if we look really carefully, the only difference is that CH3. Thymine just has a methyl, uracil doesn't. It still has the same hydrogen bonding pattern, so you get A and U, G and C when you're doing RNA, where you get A and T, G and C when you're doing DNA. What molecule pairs up with, I mean, it's basically identical except for that methyl. But it makes its stability a little bit different, and it makes how molecules fit together slightly different. And additionally, ribonucleic acid is not deoxy. So your ribose still has that extra OH. Oops. Phosphate. Oh, phosphate. Base. Your RNA has that extra OH. This changes the shape a little bit, changes interactions. RNA tends to be, you just make single strands of it. It doesn't quite fit together like DNA does. It's less stable, but it's got the same order. So what'll happen is you'll have a strand of DNA, it'll open, and it'll curl up. But here, for a little bit, you'll have A, T, C, C, G, A. You'll have a string of your bases, and then there'll be the matching string T, A, G, G, C, T. So one strand will have one of the two, the other will have the other of the pair. So A and T, T and A, C, G, G, C, G, T. Well, you can unravel the DNA a little bit, and then what will happen is you will have transcriptases that come in and read it, and they will create RNA to match it. And they'll say, hey, okay, what do I need? I need, what's opposite T? Well, that's A. What's opposite A? Well, it would be T, but in RNA, uracil replaces thymine. So TA still pair, but it's just U for the T when it's in RNA. G and C still pair, C and G still pair, T and A still pair. What you've done is you've created a strand that if we want to, we take a second and kind of bring up and compare here, looks really identical to the first strand. The only difference is anywhere there was a T has been replaced by a U. So we've created what's effectively a copy of the original strand of DNA, just made it out of RNA. Now, normally in a cell, eukaryotes at least, we have our nucleus, our DNA hangs out in here where it's small and protected, and then RNA will leave. Once we've copied the section we need, it goes out to the rest of your cell, to the cytoplasm. Out here, all the things that could damage molecules are hanging out, and so our DNA stays in this nice protective area, sends the copy out. The copy runs into things like ribosomes. A ribosome reads the RNA and turns it into a protein. So ribosomes are these very large complexes, and I mean, describing what's in them, I'd say go read the Wikipedia page, but it's massive. They are multiple proteins. There are chunks of RNA permanently intertwined. There's lots of ions. These are giant molecular machines whose whole job is to take this strand of RNA and hold on to it. And so we will have A, U, C, C, G, A. There we go. So our strand drifts in. Notice I flipped it over, and what will happen is it will just hold it in place. There are other RNA molecules. And so what we've created so far was an RNA that left the nucleus. This is technically mRNA, messenger RNA. It transmits the instructions out of the nucleus into the rest of the cell. There are a few types of RNA that are constructed elsewhere, and these are tRNA, transcription RNA. These are chunks of RNA who fold up on themselves and look like this. And so there's A's and U's and C's and G's. Like, they fold up on themselves. Attached here is an amino acid. Serine, alanine, lysine, something. There's a protein out there that grabs tRNA, slaps an amino acid on it. The difference is, how do you know which amino acid put on? At the very bottom will be a 3-letter code. And so this code would be something like G, A, U. This just hangs out on the bottom, which means if I have a large protein, well, I can have a protein wrap around it and have the right R groups such that they bind really well to that particular pattern. And they also bind pretty well to that amino acid. So we're actually able to construct this, like this free-floating transcription RNA that doesn't have the amino acid. It locks into this protein, some enzyme, and then the enzyme slaps the amino acid group into it. tRNA then leaves, says, hey, I'm out of here, I'm done, drifts away. Protein is somewhere else at this point. This tRNA, though, a little bit big at the moment, so I can shrink it. What will happen is it will drift along until it finds the RNA, sorry, the ribose, and the other RNA, and it locks into an active site in the ribosome. So the ribosome captures it, and it pops in and out. It goes in, it goes out. It goes in, it goes out. It doesn't hold particularly well. But if your RNA is in there and has just the right matching letters, it gives a little extra grip. It holds on a little bit more and can trap it. At this point, it gets stuck in the ribosome, and what will happen is our RNA, our messenger RNA, will then shift. Our tRNA shifts over. What will happen is another tRNA with a separate amino acid 2, and different codes. So this one would be UCG. Eventually, this other tRNA will drift by, bounce in and out, and if it's the right match, which in this case G to C, C to G, A to U, what will happen is they will line up together. The amino acid A, or the first amino acid, jumps off onto the second one. And what ends up happening then is you link your amino acids. Amino acid 1 goes to amino acid 2. And this just keeps happening. You keep going down your RNA. Sorry, green. So all your other letters you might have, GG, or GC, U, AAA, over and over and over. The RNA will shift. A new tRNA will eventually stick. The amino acids will link together. The result is you get a specific set of amino acids. If you want alanine, serine, histidine. The idea is there is some 3-letter code. I don't remember the codes off the top of my head, but we'll say X, Y, Z, ZZZ, XXQ. Right? So these would really be A's, T's, C's, and G's. I just don't remember the 3-letter codes off the top of my head. The idea is your RNA first 3 would come in, the matching tRNA with alanine would stick to them. You'd put alanine in your first. Your second set of 3 of your RNA would get moved up. tRNAs would keep bumping into it until one stuck. Only serine is correctly matched for it. So the tRNA with serine would then attach. You'd then move to your third set of 3. tRNAs would bounce into it over and over until the one that matched, in this case histidine, stuck to it. Histidine would get attached. And this would just keep happening over and over and over for as long as you had more RNA. And this is how we actually build proteins. Those proteins, those small peptides, those giant 60,000 unit, or sorry, 65,000 grams per mole was hemoglobin. So tens of thousands of amino acids long. Right? These are constructed by reading that RNA 3 bits at a time. And those 3 bits are called codons. The original set from DNA is a codon. So the mRNA has a codon. The anticodon is on the tRNA. And so our original ones had been A, U, and C. And so these 3, the matching set would have been G, A, and U. And so your codon was the original 3-letter code from the mRNA. The anticodon is the matching 3-letter code from the tRNA. And so by simply matching up the opposite letters, the tRNA that has the appropriate amino acid attached drifts in, because that 3-letter anticodon on the tRNA, every single one of those tRNAs with that combination has the same amino acid. Amino acids, because the proteins that connect them are very specific, they will only attach serine to something with the right 3-letter codon. They won't put it on any other codes. Now, you can have multiple codes that are serine, but you will never have the same code have 2 different amino acids on it. And so this is the basic structure and the basic function of DNA, at least and from the chemical viewpoint. Most likely you've had some bio classes that may have gone into this a little bit deeper in terms of the biology components of it, but as a chemical standpoint, from the shape of the molecules, the structure, and the nature of the interaction that allows them to turn a chunk of DNA, using its matching bases to create your RNA, and then using your RNA's codons and the matching bases to create your protein by matching your anticodon. These physical interactions and these shapes, this is how it's accomplished. So this is what's going on to translate DNA.